Welcome everyone. In this lecture, we will discuss the next topic in mineral nutrition, that is mechanism of absorption of elements. In NCERT, under topic 12.3, they have given mechanism of absorption of minerals, right? Or elements. Means we are going to understand how the plant will absorb minerals from the soil. Okay. Now, if we have to understand the mechanism, means we have to conduct a study, right? So, how can you conduct a study? Can you conduct a study with the soil? Can we control the soil parameters? Can you control the soil nutrients? No, right? So that is why the study on mechanism of absorption of elements was conducted on isolated cells. In lab you will do it. You will take a cell and you will see how is the ion coming inside, or you will take a plant tissue and you will see epithelial tissue or mesophyll tissue. You will take and you will see how the minerals are coming inside, or you will take an organ. Root is an organ, stem is an organ, leaf is an organ. You will take an organ and you will see how is the absorption of elements taking place. So studies on the mechanism of absorption. In NCERT, the first statement is studies on the mechanism. Studies on the mechanism of absorption of elements by plants. See, try to understand the sentence. How plants are absorbing minerals. How plants are absorbing minerals. For that, when we are conducting studies, we are conducting them in isolated cells, isolated tissues or isolated organs. Has been conducted. Where are you conducting? Has been conducted in isolated cells. We are conducting it on a isolated cell, or we are conducting on a tissue, or we are conducting in an organ. We will take a cell, or we will take a tissue, or we will take an organ, and then we are seeing how is the plant cell absorbing minerals, by which process it is absorbing minerals. So this is the first point. Understood? So when we have to see the mechanism of absorption, then you have to conduct a study. For conducting studies, you cannot go into the agricultural land and conduct, because soil you cannot control. That is why you are doing studies in the lab by taking an isolated cell, right, or an isolated tissue or an isolated organ. Now, after doing these studies, the studies revealed that there are two phases of absorption. So, in NCERT, the second point is these studies revealed that there are two phases, or there are two phases. The studies have revealed that there are two phases. First one is the initial phase, next one is the second phase. Let us see what are the two phases. Now, they told first one is the first phase and next one is called as second phase. Next one is called as second phase. Now, for example, if this is the root and this is the root hair and this is the surrounding soil. Now, from the soil, the minerals are coming into the spaces. It is coming into, right, into the space the mineral is coming. So, the first phase is rapid movement. From morning to evening, you did not water your pot, so it is completely dry. The root is completely dry. The moment you add water to the soil, the root initially it takes water very fast. So the initial phase of water absorption and mineral absorption is very rapid. So we can tell in the first phase, the absorption of elements. The absorption of elements is rapid. The author is telling it's very fast. The absorption of elements into the plant is very rapid. And where is that water and minerals going? The ions are going into the free spaces. From the soil, they are going into these free spaces. It is rapid and ions move. Where are they moving? They are moving into the free spaces. They are moving into free spaces. From outside, the ions are getting into free spaces. Now, what are the points we can tell in this phase? Now, since the movement is happening across a cell wall which is dead and they are coming into free gaps, they're not entering the cell, they're coming into free gaps, so it's called apoplast. So, the movement is called apoplast. And then we are telling it is apoplast and it is coming rapid movement. We wrote here. Where did we write? Yeah, it is a rapid movement. Means it is a fast movement, we can tell. And the particles are coming from high concentration to low concentration. Means along the concentration gradient. Ions are moving along the concentration gradient. From high concentration to low concentration they are coming, and the movement is rapid or it is fast. For example, if we have to understand, we will draw. This is one cell, another cell and another cell. Now their walls, the cell wall will be like this, right? Now this is the wall. When the water is entering in from outside, the water, it passes through these free spaces. Through the cell wall it is going. Through the cell wall it is going. So, passing water or passing minerals through the wall which is dead is called apoplast. So, it is apoplast, and since through the spaces it is passing, it will be fast. And from high concentration to low concentration it's coming. It is along the concentration gradient, and we will write down it is passive transport. It is passive transport. Means, what do you mean by passive transport? No energy requirement, no energy expenditure. This is the first phase of mineral absorption by the plant. Let's see again. The NCERT says that it occurs in two phases. The first phase is very rapid. The minerals are coming into the spaces. Means it is in this manner. Through the walls it is moving. So since it is moving into the free spaces through the dead wall, the mineral transport is apoplast. And through the spaces, since it is moving, there's nothing to restrict it. So the movement will be rapid and fast. And it's always from high concentration to low concentration, means it's along the concentration gradient. And such a type of transport will be passive transport. Now let us talk about the second phase. In second phase, the ions will move into the cells. They will cross the cell membrane. In NCERT, they have given the second phase. In second phase, ions move into the cells. The ions move into the cells. If they have to enter into the cell, means it has to cross the membrane, right? If we have to draw the picture again to tell this transport, right? So now this is the wall. Now here the ions are moving into the cell, and from here they are moving into this cell. From here they are moving into this cell. They are crossing. If this is cell wall, they are also crossing. This is the green color one is cell membrane. Whenever it is crossing a cell membrane, the process becomes slow. So that means if this process is rapid, this process is slow. If this process is apoplast, since it is moving through the gaps, this process is symplast, since it is moving into the living tissue. And here it is along the concentration gradient, from high concentration to low concentration. But we know that the cells will have the mineral. So means from low concentration to high concentration, means against the concentration gradient. And here, because it is moving from high concentration to low concentration, it is passive transport. Energy is not required. Since cell is already having the minerals, from less concentration it is coming to high concentration. With the expenditure of energy it will come, means it is active transport. So in the second phase they told that it is symplast. So let me write down here. Here I wrote apoplast. That means here we need to write it as symplast. And here we wrote rapid or fast movement. Here we need to write it is a slow movement. And here we told from high concentration to low concentration. So it's along the concentration gradient. But it is from low concentration to high concentration. So I need to write it is against the concentration gradient. It is against the concentration gradient. So if it is against the concentration gradient, then we need to expenditure of energy. We need to spend energy. Then it will not be a passive process. It's going to be an active process. So it is an active process. Active process means energy requirement. Energy requirement is there, and these ions are moving with the help of pumps, with the help of transmembrane proteins, we can tell. So these ions, how are they moving? On the membrane, there are some transmembrane carriers. There are some specific carriers. With the help of the carriers, they are moving. These ions move with the help of special pumps. Active transport uses a special pump. Take a screenshot. We will continue. Right, children? Did you understand? Can I erase this? This is what is the explanation for the 2-3 points what they have given in the initial lines of this topic. 12.3 mechanism of absorption of minerals by the plant. Now, how do you came to know that the second phase of absorption is active transport? Now, my question is, how do we came to know? How do we came to know that the second phase is active transport? How do we came to know that the second phase is active transport? Let us try to understand this question. There is an aquatic plant called Nitella. There is an aquatic plant called Nitella. This Nitella, it absorbs more of K+, more of phosphate ions from the pond water. How many times it will absorb means very greater concentration than the pond water. Means pond water has less K+, that K+ also Nitella will take it. Okay, so this experiment tells that from low concentration to high concentration it is coming, and we know it is against means we need to spend energy. So evidence we can give Nitella plant. What is it? It is an aquatic plant. Nitella absorbs. What does it absorb? It absorbs K+ and phosphate ion. It is absorbing K+ ions and phosphate ions. So many times it absorbs many times greater than the pond water concentration. Absorbs K+ and phosphate ion many times greater than pond water. Means from the water, whatever K+ and phosphate ion concentration is there, the plant is taking it off. Means from low concentration to high concentration it is coming. If it is going from low to high, it is against. Whenever it is against, then it requires ATP. Now from where do we get ATP? From where do we get ATP? For example, if I am working, I will get my salary, that is my money. In the same manner, how will cell get currency? How will cell get ATP? By doing respiration. Now, try to give some inhibitors, metabolic inhibitors, and stop the path of respiration. Hold on respiration, stop respiration. If you stop respiration, no ATP generation. If there is no ATP generation, then Nitella cannot take K+ and phosphate. A mineral accumulation is not there. So by doing this experiment also, we can tell that like Symplast is an active process. So another experimental evidence. We got it. From where did we get the another experimental evidence? By using metabolic inhibitors. What is that metabolic inhibitor? It blocks respiratory pathway. Metabolic inhibitor which blocks respiratory pathway. It stops respiratory pathway. Now, what is an example for such metabolic inhibitor? Azide is one example, right? Azide is one example. Cyanide is another example. Amma, if we give azides and cyanides, then respiration will not happen. If respiration is not happening, no ATP generation. If no currency is there, if no ATP is there, no active transport. No active transport. If no active transport, no accumulation of ions in the plant cell. So then there is no accumulation of ions inside plant cell. Understood, children? So if I ask you, like, how do we came to know that the second phase of mineral absorption is active process? So if we check a Nutella plant, in Nutella plant, if you see the K+ concentration and phosphate concentration, it is far high when compared to the surrounding pond water. Means this is understood that plant is taking more minerals. Means from low concentration to high concentration means it is active transport only. One more experiment we did using metabolic inhibitors like azides and cyanides. If we use, they stop respiration. When respiration is stopped, then no ATP generation. When no ATP, then what happens? No active transport. When there is no active transport, the Nutella plant is not having minerals. Understood, children? Understood till here? So we understood the first phase of mineral absorption. We understood the second phase of mineral absorption also. And then in NCERT they tell that like movement of ions across the membrane. The simplest movement of ions across the movement is called as flux. We use a word called flux. Movement of ions across the membrane. If you are moving ions across the membrane, then we use a word called flux. Now, if we take a cell membrane is made up of phospholipids, right? Phospholipids has heads and tails, right? So I'm trying to draw head here, tail, head and tail, head and tail, right? So I'm drawing a cell membrane, right? Good. Now, through this cell membrane, now this is inside and this is outside. Now, if the ion is entering in, if the ion is entering in, right? For example, if K+ ion is coming in, that's called influx of K+. What is it called? It is called influx of K+ ion. And if any ion is going from inside to outside, if any ion is coming from inside to outside, let us take sodium. Sodium if it is moving outside the cell, sodium if it is coming this side, right? So if any ion is coming outside, then that is called efflux of Na+ ion. It's called efflux of Na+ ion. In NCERT, this much information is there about 12.3 topic, mechanism of absorption of minerals. First, they told like studies when they have conducted, they conducted in isolated cells, not in the farmlands. The second point they told, these studies revealed that the mineral absorption occurs in two phases. First phase they told, it is rapid movement, it is into the spaces and it is apoplast without energy, it is passive transport they told. The second movement is Symplast they told, it is slow they told, with the expenditure of energy with the help of pumps they told. Now how do you come to know that the second phase is active? Then we did these two experiments to tell that yes, the second phase of mineral ion transport is active. Then afterwards the NCERT says the movement of ions across the membrane inside outside, then we have to use a word called flux. If ion is coming in, influx. If ion is going out, efflux. Take a screenshot, we will continue. Right, children? Now, if we have to differentiate what is active process, what is passive process? Let us see that. So if we study active and passive process in little better manner, then I think you'll have better understanding about this concept. Okay. Now let us try to understand what are the differences between active and passive transport. Now we'll start with passive transport. Right? And active transport. Now we know that passive transport is from high concentration to low concentration, right? Passive transport is from high concentration to low concentration, right? So then it is along the concentration gradient. What word we use? It is along the concentration gradient, right? So since it is going from high to low, no energy expenditure, no ATP expenditure, right? Whereas in active transport, it is going from low concentration to high concentration. From low to high, we need to send the particle. So means along the concentration, against, it is against. So write down, it is against the concentration gradient. And then if it is moving from against the concentration, from low to high, with ATP consumption. So we can tell with ATP expenditure. It consumes ATP, breakdown of ATP is required there. Now, if we have to see how many types of passive transport are there, there are 3 types of passive transport. One type of passive transport is called as mass flow. Mass flow of ions. One type of transport is called, passive transport is called mass flow of ions. In mass flow, see, particles are moving in mass across a membrane. In mass flow, what happens? Particles are moving, ions are moving in mass. Ions are moving in mass. From high to low, that is called mass flow. And what is the second type of passive transport? It is called ion exchanger transport. What's the second type, Amma? It is called ion exchanger transport. Now let us see what is ion exchanger transport. Let us draw a route. Let's draw a route and let us take a soil particle. So these are soil particles, right? Now the root, right? What the root is doing? So root has root here. From this, the H plus particles are rotating. These are the H plus particles. They are rotating. They are coming back. In the same manner, soil also is having particles like K plus if it is there. Now on the soil, again K plus particles are there. Now this is K plus. Now these K plus particles are going in this manner. Now if you see here, there is a point of collision. There is a point of collision. When both are colliding with each other, what happens? H plus move this side and K plus comes this side. So now K plus should go this side to the soil, but K plus is coming this side. And H plus should come to the root. This is root. H plus should come to the root, but H plus is going to the soil. K plus, they exchange, right? The H plus which is with the root, the K plus which is with the soil, when they are moving around themselves at the point of collision, they got exchanged. H plus is going this way, K plus is coming this side. This is called ion exchange transport, which is again a type of passive transport. One more ion exchange is also there, bicarbonate transport. Okay, so I will continue it here. So with this, the active transport is over. We'll see it in the next board. So we'll talk about the next type of ion exchange transport, right? So that is bicarbonate ion transport, another mechanism. In bicarbonate ion transport, what happens? Again, let us take the root. Now, root, when it will do transpiration, plant when it will do transpiration, it releases carbon dioxide like us only. Now, the surrounding is the soil. Now, soil has water. Now, this carbon dioxide molecule combines with water. Carbon dioxide plus water, it will make carbonic acid. This carbonic acid, it will break down and it will make H plus ion and bicarbonate ion. It makes H plus ion and bicarbonate ion. And here we have soil particles. Now we know that soil particles are colloided with K plus particles. Now in the soil, what happens? K plus particles are moving. That K plus combines with H plus. So when H plus combines with K plus, potassium bicarbonate is formed. A salt is formed. What is that? It is potassium bicarbonate. This potassium bicarbonate salt is taken by the root. This potassium bicarbonate salt is taken by the root. So we are just checking in how many ways the plant is taking the minerals. One way we told passive, another way we told active. In passive, if the particles are going like masses, 1, 2, 3, 4, 5, 6, 7, like this they are going, then that is called mass flow movement of ions. If they are exchanging, soil has K plus, root has H plus, so H plus going to the soil, K plus coming to the root, it is called ion exchange mechanism. Another way of ion exchange is when this carbon dioxide from the root respiration, when it comes out, when that combines with water molecule, it makes carbonic acid. That carbonic acid splits into bicarbonate ion and H plus ion. This bicarbonate ion, it takes the K plus particle and it makes potassium bicarbonate salt. That salt form is taken by the root. This is another type of transport. Now take a screenshot. We will continue further. Right, now we are going to talk about the third type of ion exchange transport, which also comes in passive transport only. We'll talk about the third type of ion transport, which is called Donand's equilibrium. Right, now this Donand's equilibrium is due to the movement of, or is due to the presence of non-diffusible ions. Donand's equilibrium is due to the presence of non-diffusible ions. For example, let us take a soil particle. Now this soil particle, right, and this is root. This soil particle has some negative charge. It has some negative charge. Because of this negative charge, this is a non-diffusible ion. What is this? This is non-diffusible anion. It cannot come out. Because of this non-diffusible anion, because the concentration is more, the positive charge from outside moves inside. Which charge is moving? Positive charge. How much positive charge will move? Till it balances the negative charge. So, one positive charge will move, second positive charge will move, third positive charge will move, one more positive charge will move. So, because of the presence of this non-diffusible anion, so cations started coming in. Not only here, in the root also the same situation will be there. In the root also the same situation will be there. Now, in root, if non-diffusible cations are there. In root, if non-diffusible cations are there, because of this non-diffusible cations, they cannot go out. Now what happens? Negative charge starts coming in. How many negative charge ion particles will come? Till they balance with this positive charge. Now this is what is equilibrium. In Donand's equilibrium, we can write a statement like the cations which are outside and the anions which are outside, so they balance with the cations which are inside to the anions which are inside. So that is at equilibrium. So if you see outside, we need to draw only 4 here. If you see here, there are 4 cations, 4 anions. Here also there are 4 cations, 4 anions. This equilibrium is called as Donand's equilibrium. And remember, Donand's equilibrium is because of non-diffusible ions. It is because of non-diffusible ions. Children, all this is about passive transport. In passive transport, we told movement can be in mass, or movement can be with the help of ion exchanger. In ion exchanger, we talked about the first one. What did we say? It is K plus and H plus exchange. The second one we talked about bicarbonate exchange, and third one is Donand's equilibrium. All this is about passive transport. Take a screenshot, then we will continue with the mechanism of active transport. Right. Now let us talk about the mechanism of active transport also. Now we know active transport will be from low concentration to high concentration with the expenditure of energy and through the membrane, and it involves special carrier proteins, right? So let us draw head and tail, again phospholipid head and tail, again phospholipid head and tail. I am drawing a membrane. Right, children? And we need to, because cell membrane is bilayered, so we need to draw one more membrane here. Right? So we are drawing one more membrane here. And the spaces which I have left, I'm going to fill it with some special proteins. I'm going to fill it with some special proteins. For example, we'll take a protein here. This is one protein, and we'll take another protein here. Right? We'll take another protein here. So this is outside of the cell, and this is inside of the cell. This is inside of the cell. If we check outside, the concentration, we are talking about active transport. We are talking about active transport, children. Now if you see H plus ion concentration, usually it will be more on the outside only. H plus particles, I am keeping them more on the outside. 4 H plus particles are there. There is only 1 or 2 H plus particles inside. But still this H plus particles want to go outside. Means from low concentration to high concentration they are going. From low concentration to high concentration, the H plus particle wants to go. Now if it wants to go, it is active transport, it needs to spend energy. Currency of the cell is ATP. Then what happens? 1 ATP gets hydrolyzed. 1 ATP gets hydrolyzed. It is broken to an ADP molecule and phosphate molecule. When you break the bond, you get the energy. Using this energy, the proton will be carried across this carrier pump from inside to outside. Inside is called Symplast. Inside is called Symplast. And outside will be called space. No, it is. So we can call it as Apoplast. So with the hydrolysis of ATP molecule, what happened? Proton moved from Symplast to Apoplast. When all the protons are moving this side, when these two protons move from this side to this side with spending ATP molecule, what happens? Proton gradient will be built up, right? Because this proton molecule went here. This proton molecule also, when it goes here, proton gradient will be built up towards the Symplast. So all the protons which are outside, they want to return back. They have moved from here to here by spending ATP. They want to move back. How can they move back? By creating a proton gradient. What is generated here? Proton gradient. Proton gradient is towards the Symplast. Protons want to move back again. Now, if they want to move back again, from the place they entered, they cannot come. So, they will check for another carrier protein which is on the same membrane away from this one. Okay. Now, if you see this protein through this path, the H plus particles are trying to come in. The H plus particles are trying to come in. They will come back inside. Now, when the H plus particles are trying to come back inside, along with H plus particles, if it's a plant cell, right? Sucrose molecules, if they are also patching and they are coming. Sucrose molecule, it will tell, like when you are going, take me also in. So, if the sucrose molecule is also trying to come inside, if the sucrose molecule is also trying to come inside, along with the H plus particle, what is this, ma? This is sucrose. If this is also trying to come inside, then this is called active transport. What is this active transport? What is this active transport? This active transport, when H plus moved out, is called as primary active transport. This is primary active transport. Because of the proton gradient, what happens? Proton motive force get generated. We can tell PMF. Because of more protons here, proton motive force is generated. Using that force, protons are trying to come back inside from apoplast to Symplast when they are coming. Along with that H plus particle, if another solute also tries to come inside, then that is called secondary active transport. What is this called? This is called secondary active transport. Children, notice one point here. This 100% everyone will agree that it is active transport because ATP is used. ATP is hydrolyzed. But here there is no hydrolysis of ATP. Then don't get confused. Don't think this is passive transport. It is active transport. If this is not happening, will the situation come? If the protons are not going out, will the protons come back in? No, right? Because of the proton gradient only, because of the proton motive force only, the protons are coming in, bringing sucrose molecules are also coming in. So that is why we call this as secondary active transport. And where the ATP is hydrolyzed is called as primary active transport. Understood? Now, if two particles across this carrier protein, what is this? This is a carrier protein, right? If two particles across a carrier protein are coming in the same direction, then what is it called? Symport. So this is an example for symport also. Why are we calling it a symport? Because two particles in the same direction they are coming. And see here, only one particle is moving. That means this is uniport. If only one particle moves, that is uniport. If two particles are moving, okay, then if two particles are moving, you can call it a symport, which is a co-transport, right? Two are moving, so you can also call it as co-transport. Co-transport, both the particles in the same direction, it is symport. If at all, if one particle comes in and the other particle goes out, then that is antiport. If one comes in and the other goes out, what is that called? That is called antiport. Children, I think you understood what is this. You understood what is active transport mechanism and what are pumps and how is ATP hydrolyzed, how is the proton gradient build up. You understood, right? Now, if we have to give examples for symport and antiport, so I will give examples for symport here. We will take examples for symport here. Symport example just now we saw H plus with sucrose is an example for symport. In the same manner, H plus with amino acids. Like sugar, amino acids also if they come inside, that is also example for symport. And if H plus Cl ions are also coming inside, if both are moving in the same direction, symport. Antiport, if we have to give examples, we can tell if H plus is coming in, Na plus will go out. If H plus is coming in, Na plus will go out, that is antipode. And if H plus is coming in and Ca plus 2 is going out, that is antipode. So this and this are examples for antipode. So 1, 2, 3 are examples for sympode, and what we have seen on the board is about active transport. I think with this lecture you understood the mechanism of absorption of elements by plants. What is the first? What is Symplast? What is passive transport? What is active transport? What is uniport? What is co-transport? What is primary active transport? What is secondary active transport? Now in the next lecture, we'll try to start nitrogen metabolism and nitrogen cycle. If you like the content, like, share and subscribe the channel with your friends. Thank you.